going to be an overview of glycogen metabolism and how our bodies regulate our blood sugar, so our blood glucose levels. But more detail on all of these different regulatory steps in the past video I'll link to, but today I just want to kind of give the big picture overview, more of a review probably because it's complicated and I'm not going to go into all of the details and it looks really scary, but it's actually really logical. And so I'm going to show you some of this logic, how it comes about, and then I'll show you some actual data from some metabolic tracking studies where they show what happened to hormone levels and what happened to blood glucose levels and tissue glucose levels when somebody ingested sugar. So our sugar in the body is our glucose. That's our main sugar that our bodies use, this monosaccharide. But instead of storing glucose as individual glucose units, our bodies link them all up into glycogen and store it as glycogen. Now, this isn't in all of your tissues. So basically your liver and your muscles are the main places that glycogen is stored, but they're not the only tissues that need glucose. So for example, although most of your tissues actually rely on free fatty acids or FFAs for their main source of energy, some tissues like your brain and your erythrocytes, so your red blood cells, well, your red blood cells don't even have mitochondria, so they can't do all that beta oxidation, that good breakdown of those fatty acids to get energy. And so instead, they rely exclusively on glycolysis. Your brain also really relies on glycolysis, and it can't store its own glycogen. Bottom line, we need ways in which we can provide glucose to body tissues in need, such as your brain, such as your red blood cells, and such as your muscle cells when they run out of their glycogen. But your muscle cells are able to hold glycogen as well as your liver. Now, your liver works as a great team with your pancreas in order to really control your blood sugar levels. So your liver and your pancreas are going to be the main players there. Your pancreas is responsible for sending out the hormones. So we'll talk about how it sends out glucagon as well as insulin. And so glucagon that says, hey, the glucose is gone. Let's send out some glucose, and the insulin says, well, there's a lot of the glucose is high. Let's let some glucose in. And so we'll talk a little bit more about that in a second, but those messages are coming from your pancreas and getting received by various tissues. Now, when the liver gets that signal, it's going to make the liver start kind of either breaking down glycogen, so glycogenolysis, or it's going to start building glycogen, so glycogenesis. So there's a lot of G terms and don't confuse them. Glycolysis, this is the breakdown of glucose into pyruvate. Gluconeogenesis, that's the making of glucose from non-carbs, so like our pyruvate. This is not what we're talking about here. Instead, we're talking about glycogenesis, so the making of glycogen from glucose, so linking together those individual glucoses, and glycogenolysis, so breaking down that glycogen to give us glucose. Well, a little bit of a complication is that what you end up with is actually glucose 1-phosphate, so a phosphorylated form of glucose. And the only tissues that are able to actually dephosphorylate that glucose are going to be your liver and your kidney. So only the liver and the kidney are actually able to, either from glycogenolysis or from gluconeogenesis, so the fresh making of sugar, only they have this glucose 6-phosphatase that they can use to remove the phosphate group and therefore generate free glucose that can get shipped out to your body. So only your liver and kidneys are able to actually provide glucose that's freshly made or freshly freed to other tissues in need. Yes, and I know that I said glucose 1-phosphate was released, and it is, but that can be interconverted through phosphoglucomutase to give us our glucose 6-phosphate, which can then be through glucose 6-phosphatase, which is, remember, in the liver and kidneys, that can get freed up into glucose, which can be released. Now, remember that I said that the liver and the muscles were the main tissues where we had our glycogen stored. If we break down glycogen in our muscles, well, that's glycogenolysis. And well, there we would end up with glucose 1-phosphate. We could convert that into glucose 6-phosphate, and we could use that for glycolysis, burn it to get ATP, Or we could also take it in various other pathways. If we wanted, say, glycosylation precursors, we could go the hexosamine pathway. If we wanted NADPH or nucleotides, we could take it down the pentose phosphate pathway. But we could not make it into free glucose, and therefore we cannot release that glucose into the bloodstream. So the muscles get to keep it all for themselves. Remember, our cells have glucose transporters, but we do not have glucose 6-phosphate transporters or glucose 1-phosphate transporters in our plasma membrane, and so we can't ship out that phosphorylated glucose. Muscles get to keep it. What about the situation in our liver? Well, in our liver, if we break down that glycogen, remember that's glycogenolysis, we get our glucose 1-phosphate, we convert that to glucose 6-phosphate. Well, now we can actually dephosphorylate it and we can ship it out. What about our kidneys? Well, our kidneys don't actually store glycogen, but they can make glucose from glucagenesis. So remember, that's when we're making glucose from non-carbohydrate precursors, such as our pyruvate. We can take our alanine, we can take lactate, convert these into our glucose 6-phosphate, and then our glucose, because those tissues have that glucose 6-phosphatase. So your liver and your kidneys can make glucose that then they can ship out to your bloodstream. So the livers and the kidneys are both able to provide glucose to tissues in need. And we'll see when I show you some of that actual metabolic tracing data that your liver provides about 80% of the glucose in your bloodstream, the free glucose that's like shipped out by your body, and about 20% of it comes from your kidneys. The stuff that's coming from your kidneys is just from gluconeogenesis, but the stuff that comes from your liver can come from gluconeogenesis or from glycogenolysis. And which one it is depends on, well, like how soon after have you depleted your glycogen stores. So your liver stores a lot of glycogen, but it'll run out of it. And once it runs out of it, well, now the only way that it can provide glucose to tissues is through gluconeogenesis. Thankfully, your tissues are sending it a bunch of alanine and lactate, as we'll look at, and the liver has a bunch of fat it can break down and get the glycerol from that to make glucose. So different ways in which the liver can then go and provide glucose to the body. So key points about tissue-specific roles in glucose metabolism. We have our liver and muscles are the main places that glycogen is stored. Remember that the process of making glycogen is called glycogenesis. So genesis make, glycogen, well, that's what we're making. So the liver and the muscles are going to do a lot of glycogenesis in response to elevated blood sugar. And the hormone that's released in response to elevated blood sugar is going to be insulin. And so we'll look and see how insulin is actually able to make these changes come about. Your liver and your muscles, if they need energy, well, now they can break down that glycogen. That's called glycogenolysis. The liver does it to ship glucose to the tissues, and the liver basically, it'll just say, okay, you guys can have this glucose and I'll rely on fats for myself. Whereas the muscle's like, no, I'm not shipping out this glucose. I can't even take the phosphate group off, so I'm just going to use this glucose 6-phosphate for myself, take it down in glycolysis. I can either break it down all the way through oxidative, get all the way to like oxidative it fully and do the TCA and the ETC and get all that ATP. Or if there's not enough oxygen or I'm just lazy, I can take it just to pyruvate, take that pyruvate to lactate or alanine, ship those to the liver, which likes that likes the lactate, or the kidneys, which like that, they like the glutamine, and well, the liver likes the lactate and the alanine, and your kidneys like the glutamine more. But basically, there are different ways in which you could then ship those carbon skeletons to those kidney and the liver in order to make fresh glucose for them. Okay. Then the muscles basically break down the glycogen. They use the glucose 6-phosphate. The liver breaks down that glycogen, takes it to glucose, and ships that out. If there's not enough glycogen and the blood sugar is low, both the liver and the kidney can make glucose in the process of gluconeogenesis and then ship those out. Now that we have that idea, let's look at how this actually goes about. As I mentioned, there's going to be a couple of main hormones, and these hormones are going to be secreted by our pancreas. So there are multiple types of cells in your pancreas, and they secrete different hormones. So glucagon, this says all the glucose is gone, and I like to think about it with like a little alpha cell in the A in the owl, because this is released by pancreatic alpha cells. And this is released in response to low blood sugar. We'll see that it signals largely through pKa, so protein kinase A, also known as cyclic AMP-dependent protein kinase. What it's going to do, well, what do we want it to do? We want it to be able to raise the blood sugar. How it's going to do this is by promoting glucose release by the liver, and where's that glucose going to come from? It's going to come from glycogenolysis, so basically our glycogen breakdown, and it's going to come from our gluconeogenesis, so our glucose production in the liver. Apologies that this little window is here. Let me see if I can move that away. Yeah, okay. So you can glycogenolysis and gluconeogenesis. Both of those can happen in your liver, and both of those are able to then provide glucose that can be shipped out into the bloodstream. As a net result, you're going to free up or make and release glucose, and you're going to return the blood sugar to normal. What about the other situation? What if you say go too far? What if you release too much glucose? Your blood goes too high, or you just ate a big meal? Now you want to be able to kind of reverse everything. So you need a kind of opposite signal. This opposite signal is going to be our insulin. Insulin says beta lets them in. So if you think about beta lets them in, well, they're released by pancreatic beta cells in response to high blood sugar. Unlike the glucagon, which signaled through pKa, insulin is going to signal largely through pKb, also known as AKT, as well as downstream phosphoprotein phosphatase 1. Oh yeah, there's a typo there. I'll fix that. So PP1. Now PP1 is going to then be able to remove some of the phosphate groups put on by our pKa, and it's able to also regulate things in a variety of other ways. One of the ways that insulin acts is by promoting glucose uptake by muscles, heart, and fat cells. As I'll get into in a minute, it actually makes it so that they have these hormone-sensitive transporters that'll actually go to the surface of the cell and let more glucose in when they get that insulin signal. It's also going to promote glycogenesis, so the glycogen synthesis, because, well, if you've got a lot of glucose and you take in that glucose, you want to store the glucose. One reason why you want to store that glucose is, well, you don't want to have a bunch of free-floating glucose, then osmosis is going to bring in a bunch of water, cause the cells to swell and then burst. Not good. And, well, your cells probably aren't that stupid, and so, well, you have your transporters are actually going to be facilitative transporters for the most part. So they basically provide a passageway for glucose through that membrane, which is all nonpolar, and the glucose wouldn't want to go through. But basically, it's not energy requiring, and so it'll reach an equilibrium. It would reach an equilibrium if there was no changes in concentrations and things. And so what would happen was that if you have if you end up bringing in more glucose than is in the bloodstream, then things would go out of the cell, which is, well, how the liver can actually let glucose out of the cell, as well as your kidney. But if you basically link up that glucose, well, if you trap it with phosphorylation and then you link it up, well, now what's going to happen is you are going to be able to keep bringing in glucose and keep making glycogen. Okay, so that's that. Now let's go to the back to the very top, and let's look at how this all happens. There's a lot of arrows and things going on. Let's focus first on the enzymatic process, and then we'll go into the details of the regulation. I want you to follow this purple arrow and this orange arrow. The purple arrow are things that are going to be released in response to done in response to insulin, or basically things in response to a high blood sugar, where you want what do you want to do with the high blood sugar? Remember here, you're wanting to bring the glucose into the cells. You're going to want to link it up into glycogen, so that is glycogenesis. Conversely, if you want to break down glycogen, if there's low blood sugar or if there's some sort of stressor, so we'll talk about how adrenaline or epinephrine can also act in similar ways. These are going to basically promote the breakdown or the glycogenolysis. And so this is going to be these orange arrows. The core of all of this is going to be happening right in here, just these few arrows. So if we just were to look at it by itself, it wouldn't be that complicated. You take that glucose into the cells, you phosphorylate it, trap it in the cells to get you glucose 6-phosphate. Glucose 6-phosphate can go a variety of different ways. but one of the ways that it can go is through phosphoglucomutase to become glucose 1-phosphate. In order to get linked up, well, that glucose 1-phosphate, first it actually gets activated as like a sugar nucleotide. It gets a UDP stuck onto it. So like a UTP, just like an ATP, but a UTP, but only the UDP part of it. So only 2 of the phosphate groups and that uridine get actually hooked on to the glucose. This gives us UDP glucose, which has the energy we need in order for glycogen synthase to basically link them together to form glycogen, releasing the UDP. This is going to make your glycogen chain one longer. The more you do this, the lower that you continue lowering your blood sugar. You're taking the glucose in, you're trapping it through the phosphate group, you're converting it into this form that you can store it without having a big high concentration of glucose inside of your cells. Once you do this, your blood sugar is going to get lower. And well, what if you overshoot it? What if your blood sugar gets too low? Or what if you haven't eaten in a while? In this case, you're going to want to do the opposite. You're going to want to be able to take that glycogen, break it back down into glucose 1-phosphate, and then take it back to glucose 6-phosphate where you can do what you want to do with it. Now, you're going to use different enzymes to actually reverse the process. The enzyme that will reverse the process is going to be glycogen phosphorylase. Glycogen phosphorylase, don't confuse phosphorylase with phosphatase. A phosphatase is going to remove a phosphate group. A phosphorylase is going to use an inorganic phosphate in order to cleave something off. So it's able to use this inorganic phosphate to chop off a glucose 1-phosphate group. Basically, when the phosphate attacks, it gets stuck on, and so you end up with glucose 1-phosphate, which when you convert it into glucose 6-phosphate, and then if you were to go to glycolysis, well, now you've already kind of invested one of the ATP you need. However, you have to think about that you actually had to put in an extra ATP equivalent in this UTP step in order to actually link them up. So it's still more energetically expensive to make the glycogen and then break it down for the glucose than if you were to use the glucose just in the beginning. But when you go to use the glucose, well, now you don't have to invest another ATP in that hexokinase step because you already did that in the first place. So how are we going to regulate all this? Well, the first step that we could actually regulate is not even these enzymatic steps, but it's just getting the glucose into the cells. Now, there are different glucose transporters, and the amount of glucose that's actually taken into the cell, as well as released from the cell, is going to depend on the glucose concentrations, both inside and outside of the cell, as well as the transporter expression. So how much of it is made, so like the transcription and translation and all that good stuff, as well as the localization of it. So is it actually at the cell surface? And what is the affinity? How much does it like glucose? How fast can it transport glucose? All those are going to influence how much glucose is taken into the cell. You have different glucose transporters, or these like glut transporters. Your muscle, heart, and adipose tissue have an insulin sensitive one. Basically, what's going to happen is in response to insulin signaling, these glucose transporters are going to get transported. Well, that was not supposed to be a pun, but they're going to get moved to the surface of the cell. They're kind of hanging out in these intracellular vesicles, so they're already made, and then these little membrane bubbles inside the cell. And then they just kind of merge with the outer cellular membrane in response to the insulin signal. And now you have these pre-made transporters that now you can quickly mobilize to the surface and let glucose in. So now you've taken your glucose in and you're able to basically phosphorylate it. And now you go down, you go your phosphoglucomutase, you get glucose 1-phosphate, and now we go to hook it up. So remember that first step is that UDP glucose pyrophosphorylase. It makes it a nucleotide sugar, this UDP glucose, which activates it. And now we're going to reach our first really regulated step, this glycogen synthase. You'll see that here it says glycogen synthase A, and well, that A is kind of telling us this is the active form. It's a little bit confusing in the nomenclature, but there's glycogen synthase A and glycogen synthase B. These are the same enzyme, they're just in different states. So the A is dephosphorylated and active, and the B is phosphorylated and inactive. So they call it B, but really it's just the same enzyme. It's just phosphorylated and thus inactive. But don't don't confuse phosphorylated with inactive for everything. We'll see that some things are going to be phosphorylated and active, and some are going to be phosphorylated and inactive. Might seem strange, but in this way, we're able to have both phosphatases and kinases control things. So glycogen synthase A, the dephosphorylated form is going to be active, and the phosphorylated form is going to be inactive. Now we have to think about, okay, well, when would we want it to be active and when we want it to be inactive? We'd want it to be active, and if we wanted to make glycogen, and we said that that was if we had high blood sugar, we have insulin released, we want that to promote the dephosphorylation. And in fact, we see that insulin indirectly is able to basically activate phosphoprotein phosphatase 1, so our PP1, which is able to dephosphorylate the glycogen synthase, give us this active glycogen synthase, which then can make glycogen. Well, where's the phosphorylation coming from? Well, we'd want the phosphorylation to come if we had low blood sugar, or for another reason, we wanted to basically free up some glycogen, such as a response to some sort of stressor. We have some adrenaline rush. We want both of those things to be able to actually lead to the phosphorylation. And in fact, we see that one of the kinases that can phosphorylate glycogen synthase is pKa, as well as this other kinase that we'll talk about in a second, this GSK3. Now, GSK3 is glycogen synthase kinase 3. And basically what happens is that this GSK3 or the pKa, both of these can phosphorylate our glycogen synthase. And that in that way, they're going to be able to inhibit the glycogen synthase activity. We'll get back to GSK3 in a minute, but first let's focus on pKa. We'll actually see pKa in several different places in this pathway. So you can see it's coming up here, it's coming up here, and it's coming down here. So what is this pKa? As I mentioned, the other name for it is our cyclic AMP dependent protein kinase. And so cyclic AMP, remember that's the second messenger, which is released by adenylyl cyclase. So thinking back to our G protein coupled receptors, we had our G protein coupled receptors. We talked a lot about our beta adrenergic receptor. But your glucagon receptors are also able to act through a similar G protein coupled receptor and have this GS alpha subunit. Remember that S was standing for stimulatory because it could stimulate adenylyl cyclase. When adenylyl cyclase gets stimulated, it converts ATP to cyclic AMP, and that cyclic AMP can then bind to and activate pKa. Now, pKa can go and it can then phosphorylate glycogen synthase. And when glycogen synthase is phosphorylated, remember, now it's inactivated. As a result, you are not going to be making the glycogen. If we want to be making the glycogen, we'll want to dephosphorylate it. And we can do that using the phosphoprotein phosphatase that we activated by our insulin. Phosphoprotein phosphatase also gets some help from glucose 6-phosphate. So remember that we want this basically to be active when we have a lot of glucose. When we have a lot of glucose, we're going to have a lot of glucose 6-phosphate. This glucose 6-phosphate is actually going to be able to bind to this glycogen synthase, and it's going to kind of make it easier for phosphoprotein phosphatase to remove the phosphate group. And so these are going to be able to act together. So we have our allosteric regulation, our covalent regulation. This is going to help us free up the glycogen synthase in order to make the glycogen. So long story short, we have our insulin is going to promote the dephosphorylation, and we have the glucagon or the adrenaline is going to promote the phosphorylation. One note here we'll get back to in a second is that basically we saw that we had both the glucagon and the adrenaline or epinephrine could be activating that G protein coupled receptor. And you might be wondering why. Well, not all tissues are responsive to glucagon. The liver is, but say your muscle cells and your kidney cells, those are not. So if we want our muscle to be able to break down all that good glycogen that it has, we need some other signal. And well, if we think about why would we want to break down glycogen in our muscle, it's not necessarily the same reasons why we'd want to break it down in our liver. The muscle doesn't care about what the blood sugar is in terms of being able to supply glucose to the rest of the body because the muscle can't let out the glucose after it gets phosphorylated. So once that glucose gets phosphorylated in that hexokinase step, it's trapped in the muscle cells. But the muscle cells are going to want to be able to break down the glycogen they have stored in order to use it for themselves. They would want to use it for themselves. Well, if there's some sort of stressors, their fight or flight response, if you need to run away from a bear, you're going to want some energy. And if you're wanting to just move your muscles, you're also going to want some energy. And so muscle contraction is going to basically, one of the signals for it is an increase in calcium. Calcium is actually going to activate various players in the game, such as phosphorylase kinase. It's also going to have AMP that's going to act as an allosteric regulator. And so glycogen, we'll see that the muscle contraction itself is going to play a role, as well as, well, when you have your muscle movement and things like this, you're going to have some of that adrenaline, some of that epinephrine. All of those are going to help stimulate the breakdown of the glycogen. Now, I mentioned that glycogen synthase, there's these two kinases here, this pKa and this GSK3. This GSK3 is probably the one that you actually see talked about more, but it has an additional layer of regulation, so I thought I'd talk about the pKa first. But now let's look at this other complication, this GSK3. GSK3, glycogen synthase kinase 3, basically this is going to be inactivated when it's phosphorylated as well. When it's phosphorylated, it cannot phosphorylate glycogen synthase. Remember that when glycogen synthase was phosphorylated, it was inactivated. And so GSK3, when it's active, can inactivate glycogen synthase. If we want glycogen synthase to be activated, we'll want to inactivate the glycogen synthase kinase. And if we want the glycogen synthase to be inactivated, we'll want to activate the kinase. Super duper confusing, right? You got to kind of like look things over and think things over a bunch. But what would we want to promote the phosphorylation of GSK3? It'd be the opposite. We'd want to promote the phosphorylation of glycogen synthase. So in this case, our insulin is going to activate PKB, also known as AKT. This is then going to phosphorylate GSK3 and prevent GSK3 from inhibiting our glycogen synthase. Phew, okay, we got that side of things. Now let's talk about what if we want to break down glycogen. Here we need to be kind of thinking in reverse. So we've got our low blood sugar situation, or we've got some sort of stressor. We have some sort of want to move. We want to break down the glycogen, either to use it for ourselves in the case of our muscles, or to ship it out in the case of our liver cells. What we're going to do is we need to regulate this enzyme now, glycogen phosphorylase. Now, glycogen phosphorylase is also going to be activated, regulated by phosphorylation, but in this case, it's activated when it's phosphorylated. So unlike what we looked at before, which were inactivated by phosphorylation, our glycogen phosphorylase is going to be activated by phosphorylation. We have that similar weird nomenclature where we have the A form is going to be the active form, and the B form is going to be the inactive form. But here, our active form, our A form, is phosphorylated, whereas our inactive form, our B form, is dephosphorylated. So what's going to do the phosphorylation and what's going to do the dephosphorylation? The dephosphorylation is going to be done by that same phosphoprotein phosphatase we saw before, that PP1. It's able to dephosphorylate the phosphorylase and thus inactivate the phosphorylase, prevent us from breaking down glycogen, which makes sense if we have high blood sugar, if we have that insulin release, because that is when we basically want to be making glycogen and not breaking it down. And we can kind of have double duty played by this phosphoprotein phosphatase. Well, we'll actually see that's kind of got triple duty going on later. But first, let's start at this phosphorylase step. Here, it's able to inactivate the phosphorylase. So now we have to think about what is able to activate the phosphorylase. The activation happens through this phosphorylation, and the phosphorylation happens by phosphorylase kinase, aptly named. Now, phosphorylase kinase is also regulated by phosphorylation, and we also get this weird nomenclature, this A and the B. In this case, the A form is the phosphorylated form. So here, it's glycogen phosphorylase and phosphorylase kinase. Both of their active forms are phosphorylated. phosphorylase kinase is going to be phosphorylated since phosphorylase kinase is going to be phosphorylated by pKa, that same pKa that we saw before. So remember, before we saw how pKa could inactivate our glycogen synthase and thereby prevent glycogenesis. Now we're seeing that it's actually activating our phosphorylase kinase and thereby promoting glycogenolysis, so our glycogen breakdown. And so the phosphorylation of by pKa, a phosphorylase kinase activates phosphorylase kinase. Phosphorylase kinase, once activated, is able to phosphorylate and activate glycogen phosphorylase, which is then going to be able to promote the glycogen breakdown. Why these two steps? Well, the main one is like signal amplification. A kinase is able to add a phosphate group to lots and lots of different proteins, lots of copies of proteins. So basically what's going to happen is you're able to amplify the signal. You're able to phosphorylate a lot more glycogen phosphorylase than if you were just to have pKa directly phosphorylate glycogen phosphorylase. Another thing is that we can then have this extra layer of regulation where we can have signals from muscle contraction be activating our phosphorylase kinase. So when your muscles are contracting, we have calcium release as one of the main signals of it. So inside of your cells, the calcium is going to accumulate, as well as AMP. So basically you're burning ATP, you're going to end up with a bunch of AMP, and that AMP is then going to be able to serve as a signal of low energy. If you have low energy, you're going to want to free up some glucose. And so this is we want both AMP and calcium to be able to signal that we want to break down more glucose. The calcium is going to act as an allosteric modulator of our phosphorylase kinase. What it's going to do is it's going to bind to phosphorylase kinase and make it more active. So in order to have like maximal activity, it needs not only phosphorylation, but also the presence of calcium. And this is then going to be able to make it so that that phosphorylase kinase in your muscle is going to be more active. You're able to break down the glycogen. What about that AMP I talked about? Well, the AMP, that's going to stimulate our glycogen phosphorylase. And so our glycogen phosphorylase in response to this AMP is going to be more active. And basically the AMP is going to act as an allosteric regulator. It's going to bind to a site on our glycogen phosphorylase and cause it to change shape a little, become more active. And conversely, if you have a lot of ATP, you're not going to be wanting to break down glycogen. And so in that case, the ATP actually binds to the AMP binding site, but actually doesn't activate it and inhibits AMP from binding. And so it inactivates basically the protein. So we have this allosteric level of regulation on top of the level of the phosphorylation regulation. One more level of allosteric regulation we see in this pathway is in the phosphorylase kinase. It's going to be inhibited by glucose. So remember that we had basically the glycogen synthase. We saw that the glucose 6-phosphate binding was able to kind of make it easier for phosphoprotein phosphatase to do its job and dephosphorylate it. Similarly, glucose is going to bind to phosphorylase kinase, make it easier for phosphoprotein phosphatase to dephosphorylate it. But in this case, the dephosphorylation inactivates the phosphorylase kinase, and here we saw that the dephosphorylation activated the glycogen synthase. In both of these cases, the allosteric modulation makes sense. When we have a lot of glucose, or when we have a lot of glucose 6-phosphate, which we'd have if we had a lot of glucose, in these cases, we're going to be wanting to do glycogenesis, we're going to be wanting to link together glycogen, and we're going to want to be inhibiting glycogenolysis, so stop breaking it down because we've already got a bunch of glucose already. In both of these cases, the dephosphorylation promotes the glycogenesis and inhibits the glycogenolysis by making phosphoprotein phosphatase job more easy. Okay, so phosphoprotein phosphatase, we see that it's dephosphorylated our glycogen synthase and activates it. It dephosphorylates our glycogen phosphorylase and inactivates it. And it dephosphorylates our phosphorylase kinase and inactivates it. So it's able to play this like triple duty where we have all of these signals kind of be erased at the same time in response to this insulin signal. How it actually happens is a little like weird, but basically the insulin, it promotes the activation of like protein kinase B, or PKB or AKT, as well as some other kinases. Everything's not totally well understood, which is why part of why I didn't like put too much detail on here about that. But it phosphorylates like regulatory subunits that then prevents the phosphoprotein phosphatase from associating with the glycogen and things like that. But long story short, the phosphoprotein phosphatase is able to remove those phosphate groups and have the effects of promoting our glycogenesis and inhibiting glycogenolysis. So it's a really powerful molecule. And when we have a really powerful molecule, we're going to want to have something that can inhibit it. So how can we inhibit it? One of the ways is that like pKa can phosphorylate it in a place that's going to inactivate it. Another way is that pKa can phosphorylate and activate an inhibitor of our phosphoprotein phosphatase. So pKa, that good old pKa we saw before, it is able to basically phosphorylate this phosphoprotein phosphatase inhibitor 1A, PPI 1A or whatever. There's a bunch of acronyms, but basically this is going to be a little protein that binds to the PP1 and inhibits it. This protein is only able to bind to and inhibit the PP1 when it is basically phosphorylated, and that phosphorylation happens through pKa. So pKa activates the inhibitor inhibits the phosphatase, and then the phosphatase is inhibited. Well, now you're going to kind of prevent the effects of the insulin response, and therefore you're going to have the glycogenolysis be predominant. So you'll break down the glycogen. Whew, okay. So one more step in all of this that the insulin is able to do. One of the things that the insulin can do is basically promote the uptake of the glucose from the bloodstream in various tissues. We'll talk a bit in a second. I'll show you kind of like the relative affinities of the different glucose transporters. Some of your cells, like your brain cells that really, really rely on having glucose, they're going to have really high affinity glucose transporters. And they're not going to need to respond to insulin because basically they have these super high affinity transporters already. However, different cells, you'll want to be able to you want your muscles to be able to take up more glucose in response to the insulin signal. So if there's a bunch of blood sugar, you want your muscle cells to be able to take it in because your muscle cells can store it as a glycogen. Whereas your brain can't store it as glycogen, and so it doesn't make sense for it really to be responsive to that insulin. Your muscle, heart, and adipose tissue, so like your fat tissue, they all have this insulin-sensitive glucose transporter, GLUT4. This GLUT4 is kind of held captive in these little intracellular vesicles, these little membrane bubbles inside of your cell. So we pre-make this transporter, but we don't put them on the surface until we get the insulin signal. Through insulin signaling pathways, we're basically able then to bring those transporters to the surface of the cell, and then we're able to let the glucose in. When we let the glucose in, then we're able to basically start this whole process. So in this way, insulin is able to control things even before the level of all of this. You'll see that the liver is not one of those I listed that's having a GLUT4 transporter. Instead, the liver is not kind of responsive to insulin in terms of bringing in the glucose, but your liver has first dibs at the glucose, and I'll show you in a second how your liver is actually going to take up most of the glucose that actually goes through your body. And it really is this high regulator, and it doesn't need to kind of respond to insulin at that level. And we wouldn't want it to have too high of an affinity of a transporter because, well, now we want it basically to be able to let the glucose out that it makes. So let's walk through this pathway one more time, and then we'll look at that actual data. So we have a situation where we have high blood sugar. In this case, we're going to want let the blood sugar in. We're going to want to break it to build it up into glycogen that we can store. In order to bring more into the cells, we're going to have the insulin promote the bringing of GLUT4 to the cell membrane, let in more glucose. That glucose is now going to get trapped through phosphorylation. This is done by hexokinase. There's a version in our liver called glucokinase, also known as hexokinase 4. Basically, they're just isozymes, so different versions of the same enzyme. but they have different kind of regulatory properties and kinetic properties. More on that in another video. Hexokinase traps it, you get glucose 6-phosphate. You can do a bunch of things with it, but the one we're going to be focusing on now is taking it through using phosphoglucomutase in order to make glucose 1-phosphate. That glucose 1-phosphate is now ready to be taken the next step in order to make glycogen. Rather than linking it up as glucose 1-phosphate, we're going to link it up from our UDP glucose. We take UTP and we basically react it with our glucose 1-phosphate, get UDP glucose. That UDP glucose is then able to get linked up into glycogen. So this first step was done by UDP glucose pyrophosphorylase. We don't really give it that much attention because, well, it's not one of the key places of regulation. One of the key places of regulation is going to be this next step, this glycogen synthase step. Glycogen synthase is able to link together those UDP glucoses, release the UDP, and give us glycogen. End result, we took the glucose in and we stored it. Now, what if we want to basically release glucose? So either we want to release glucose 6-phosphate to use as energy in our cells from in the muscle case, or to actually release free glucose into the body, into the bloodstream, like we have in our liver case. This could be because we have our low blood sugar, we get the glucagon signal from our liver, or in our muscle cells, we've got adrenaline, we want to move, all that good stuff. Both of these cases, we're going to want to break down glycogen, that is, do glycogenolysis. We're going to want to inhibit what we did before, so we want to inhibit our glycogen synthase, and we're going to want to activate glycogen phosphorylase, which is able to break down the glycogen to give us glucose 1-phosphate. How this is going to occur is largely going to be through the actions of pKa, protein kinase A, also known as cyclic AMP-dependent protein kinase, because, well, it depends on cyclic AMP, which was going to be made by adenylyl cyclase, which was activated by our signaling G-protein coupled receptor pathways from adrenaline, also known as epinephrine, or from our glucagon. pKa is going to be phosphorylate glycogen synthase, which inactivates it, inhibiting our glycogenesis. pKa is also going to activate phosphorylase kinase, it's going to activate, phosphorylase kinase, which phosphorylates glycogen phosphorylase and activates it. Insulin is going to activate a phosphatase that's going to be able to reverse the actions of both of these. So phosphoprotein phosphatase removes the phosphorylation from our glycogen synthase and activates it. It removes the phosphorylation from our glycogen phosphorylase and inactivates it, and it removes the phosphorylation from the phosphorylator of glycogen phosphorylase, that is our phosphorylase kinase, and inactivates it. Now, pKa has a way, however, to inactivate the phosphatase. It phosphorylates this phosphoprotein phosphatase inhibitor, and that inhibits, activates the inhibitor, which inhibits the phosphoprotein phosphatase. And that way, it kind of undoes the action of the phosphatase or prevents the activity of the phosphatase. Now, insulin has another trick up its sleeve. It's going to phosphorylate PKB, also known as AKT. What AKT is going to do is it's going to phosphorylate and inactivate GSK3, which is able to inhibit glycogen synthase. So GSK3 or pKa can both inhibit this glycogen synthase, and PKB is able to inhibit the glycogen synthase kinase, this GSK3, and thereby prevent it from inhibiting the glycogen synthase. Lots and lots of layers of regulation, and not all of it is completely understood, but this is the main things that you're typically going to see. Remember that only the glucose that's made by your liver and kidneys can get released. So when I say made, it can be made fresh from like non-carbohydrate sources, in the case of gluconeogenesis, or it can be made by breaking down glycogen. So our glycogenolysis, followed by that like kind of last step in gluconeogenesis, where we have that glucose 6-phosphatase, release the phosphate group from our glucose 6-phosphate to give us free glucose, which then can pass out of the cell through the glucose transporters. And so they have lower affinity glucose transporters in our kidney and our liver, so that they can actually let the phosphate out. Let the glucose out into the bloodstream. Whereas if they had a high affinity transporter, well, now they're not going to, they're going to basically just keep bringing in the glucose that they let out. You get this like fetal cycle. Your liver and your kidney are both able to let out the glucose, but only your liver stores glycogen. So the glycogen, that the glucose that's coming from your kidneys is going to be coming from gluconeogenesis, whereas that's coming from your liver can be coming from gluconeogenesis or from glycogenolysis. In the beginning, it'll be coming, like if you just had a big meal or something, it'll be coming from our glycogenolysis. And later on, once the liver runs out of glycogen stores, then it would come from gluconeogenesis. Let's look at some actual metabolic tracking studies that show this in more detail. Or I guess put numbers to things and visuals to things. I don't know. Let's go look. So this figure's from this really cool paper I found. I found a bunch of cool papers that I'll link to. But basically what they did was they tracked some glucose after people ate glucose and tried to look and see what its various fates were. So we have our glucose uptake. So they basically ingested some glucose. Now, some of this glucose is going to go and it's going to get catabolized. It's going to go through glycolysis. And some of it is going to get stored. So this is going to be like direct storage as glycogen. What's going to happen to the stuff that went through glycolysis? Well, some of it is going to go to pyruvate, but it's not going to go through it's not going to get oxidized further. So it's not going to get converted to acetyl-CoA that can enter the TCA and then enter the electron transport chain, get oxidized all the way to give us our CO2 and our oxygen, water. So some of it is going to go this oxidative route, and some of it is going to go this non-oxidative route, where basically after pyruvate, it ends there, or gets converted to lactate or alanine that can get sent to the liver to convert it back into glucose. So some of the stuff that from this non-oxidative route, when it ends at lactate, pyruvate, or alanine, you're not just going to want to like waste that stuff. Some of it's going to get converted back into glucose by your liver. Some of that the liver is going to send out some of that through into the bloodstream, and it's going to hook up some of that to form glycogen. How much? Basically, what they found was that if they had they had 98 grams of glucose that these volunteers took in. When the glucose first goes through, you get this like splanchnic tissue disposal. So basically what this is saying is like, when you ingest things, like it goes through your like intestines and your liver kind of gets like the first dibs at it. And so this splanchnic tissue, you can think of this mainly as your liver for now. So in that kind of like first pass through you, the liver is going to take about a third of what's present. So it's going to take a big portion of the glucose before it even glucose reaches the systemic bloodstream. So before it gets to the other tissues, your liver is going to take about a quarter to a third of it. The rest is then going to go to your peripheral tissues. So it's going to get like into your systemic bloodstream. Now, what's going to happen to this? Well, basically, you're going to have some of it is going to about 2/3, I guess, is going to go through into glycolysis, whereas about a third of it is going to go to your storage. So this is going to be your initial glycogen storage, whereas the rest is then going to go into glycolysis. Most of what goes to glycolysis is going to be used in this oxidative state. So you're going to basically take it all the way down to CO2 and water. But part of it is going to go through this non-oxidative state, and so it's going to end in this lactate, pyruvate, or alanine. That can then get shipped to your liver, which can either link it up to store it as glycogen or send it back out into the bloodstream as glucose. So this route, this kind of like lactate, pyruvate, alanine to glycogen, this is like your indirect storage, and this would be your direct storage where you go straight from glucose to glycogen, as opposed to go through gluconeogenesis to glucose to glycogen. What this ends up looking like is, so you ingest this glucose, like 30, so if you had 100 grams of glucose ingested, about 30 grams is going to go direct to your liver in that kind of like first pass. What's going to happen then is that your a bunch of it is, so about 70 grams is left, goes out to your other tissues. Your brain's going to take about 15 grams of it. Your brain really needs that glycogen. Your muscle's going to take a ton of it. So your muscles is like the biggest taker after your liver, which makes sense because your muscles can store it as glycogen. Your kidney is going to take a bit of it, and your adipose tissue, so your fat tissue, is going to take a bit of it. And then you'll see that the liver is actually going to take a lot of it back already directly as is. And then it can take the products from the breakdown in other tissues. So it can take the lactate, say, that comes from your muscles, or the alanine that comes from your muscles. It can take the glutamine that comes from the kidneys actually prefer the glutamine that come from other tissues. And they can make those back through gluconeogenesis in order to give you glucose. And then that glucose can get stored as glycogen in your liver, or it can get shipped out from your kidneys or from your liver. If we look and see, okay, well, the brain took a bunch and the muscle took a bunch. Well, the muscle took a bunch and for a couple different reasons. One is that you probably, if you ate something, you would have insulin released that promotes the uptake of the glucose from your bloodstream. What about the brain? It doesn't have the insulin sensitive receptor, but your brain has this really high affinity glucose transporter. So it has, if you look at this KT, so kind of like the KM, but for like transport, it's got like a really low KT, which tells you that you have a high glucose uptake. So in our blood, normally it's about like 5 millimolar glucose. And so this is well below the level, the KT is well below that. And so it's kind of always going to be working at its maximal velocity. And you're going to be taking that glucose in, which is good because your brain isn't going to see as much glucose, and it really needs the glucose. So that's why your brain's able to take up a bunch. Your muscle, fat, and heart, they've got this GLUT4, and so they're able to basically have their activity increased by insulin, so they're going to be able to take up more as well. Okay, so yeah, and so then the liver, the liver, if we look at the liver, it's got this low affinity transporter, and this makes sense because, well, we want the liver to be able to both take in glucose and let out glucose. Additionally, if we think about the 30 grams of this sugar is going straight to your liver, because the liver's got that first pass. If the liver had that super high affinity transporter, it would basically hog all of the sugar and wouldn't let any to the other tissues, and your other tissues really need that. One last cool figure from another paper where they were tracking. Here they looked at both hormones and blood sugar levels and tissue sugar levels and stuff. What was really cool was that they were able to track they did this dual tracking, so they could track the glucose that was released from cells as well as in various tissues, as well as the glucose that was ingested. So here in this first figure on the left, they're basically showing you the different hormone levels after ingesting glucose. So initially you can see that, well, what would you expect? You would have this glucagon levels would be higher than your insulin levels because they had these people fast a little. And so they're hungry, they've got the glucagon, there's not that high blood sugar. And so you want to all the glucose be gone. But then when you take insulin, when you take glucose, well, now we've got high blood sugar. And so you see up here, we've got this increase in our plasma glucose, so our blood sugar, and we're going to want to say, hey, let some glucose in. So let's let out some insulin. The insulin, remember, is going to be released by the beta cells of your pancreas because you beta let some sugar in. Over time, however, now the sugar is going to be let into the cell. You can see that the glucose concentrations are going to decrease. As the glucose decreases, the insulin decreases as well, and the glucagon starts rising again. Now, what's going to happen to the glucose? So we said that overall, the plasma glucose is going to decrease. But you can see that some of the glucose that's in the bloodstream is going to be coming from the glucose that was just ingested, and some of it is going to be coming from the cells already, because remember that our liver and our kidneys are always going to be sending out glucose into the bloodstream. And so what they looked at here was basically in the bloodstream, how much of the glucose was from the ingested glucose and how much of it was from the endogenous glucose, so that what was already in the cells. So if we look at before they ate the glucose, you can see that most of the glucose in the bloodstream was coming from the liver, and a little bit of it was coming from your kidneys. So hepatic means liver, renal means kidneys. These black triangles are going to be your kidneys, the renal, and the white circles are going to be your hepatic, your liver. So initially, most of the glucose in your bloodstream is coming from your liver. However, you also have some coming from your kidneys. When you ingest glucose, well, now most of the glucose that's in your blood is going to be from that food you just ingested. What's going to happen to the amount that's released by your liver is that's going to decrease. There's already a bunch in your bloodstream. Your liver doesn't need to send out more. And so when we have this, the insulin is going to actually basically inhibit our glycogenolysis. It's going to inhibit our gluconeogenesis. It's going to prevent the liver from making glucose and from shipping out glucose. And so it's going to suppress those things. And so you get a decrease in the amount of glucose exported by your liver. What's kind of cool is that basically what you see is that your kidneys, somewhat surprisingly, they actually increase the export of sugar a little bit in the beginning. And they think that this might be a way to kind of like counter the decrease by your liver. So there's kind of this inverse relationship so that your liver is able to, your kidneys kind of like, I'll take over for a minute. You go and you take that glucose and you store it as glycogen, because your kidney can't store glucose as glycogen, but your liver can. And so you wouldn't want your liver to be shipping out glucose when it can store it. But if your body needs glucose, you want your kidneys to still be shipping some out. But you're not your liver. So your kidneys are going to do some gluconeogenesis and ship a little bit out, but your liver is not. Your liver is going to have it suppressed. Overall, you're going to get a big suppression of your of glucose release from the cells into the bloodstream, but there is going to be the slight increase from your kidneys, as well as a big decrease from your liver. So they were a little surprised when they found this, but they think it makes like regulatory sense that the kidneys and the liver are kind of like reciprocally regulated, so that the kidneys can kind of take over when the liver is doing its storage job. So really cool stuff, and hope you found that helpful and not just overwhelming. Yes, there's a lot of interconnected pathways and a lot of layers of regulation, but if you understand this, everything else will seem a lot more simple, and at least the logic of it is all, it all kind of makes sense. Like if you got high blood sugar, you want to do things that make it lower. If you've got low blood sugar, you want to do things that make it higher. And then it's just kind of figuring out how that actually comes about, and then how we make sure that we don't go too far one way or the other when we're trying to correct the situation. For most of us, all this goes well most of the time. And so our blood sugar levels are nicely controlled no matter how much we eat, or to some extent, if we aren't eating enough. But what happens with people with diabetes is that they have problems controlling their blood sugar, either because their cells can't produce insulin, so like their pancreas isn't producing enough of the insulin, which is what happens with type 1 diabetes, or their cells have kind of become desensitized to the insulin signal, which is the case in type 2 diabetes. So with type 1 diabetes, what happens is the pancreas, even if you eat a lot of glucose, your pancreas isn't sending out that insulin. And so this is typically like an autoimmune condition where the body's kind of like destroying the pancreatic cells, and so you don't get the insulin made. In type 2 diabetes, well, here, even though the insulin is being made, the cells are desensitized to it. And so they need, they're not kind of getting the message. Type 1 diabetes is typically treated by giving them insulin. And so because their bodies can't make enough insulin, you can give just give the insulin directly, but they need to know like how much to give. And so this is where like measuring the blood glucose levels comes into play. And so they have to regulate. So if we have high blood glucose, we're going to need more insulin. Sometimes if we overshoot things, we're going to need to take some glucose. And so that's how the type 1 diabetics are able to manage their blood sugar. In terms of type 2 diabetes, here sometimes they have insulin because basically, although their cells are desensitized to it, so they need more insulin in order to get the same signal. Sometimes supplementing the insulin can help. There are also other treatments that help in a variety of different ways, either regulating kind of various transporters, various signaling pathways, all sorts of different stuff. And there's a lot of research into what actually is at the cause of these various type 2 diabetic routes and all sorts of different things. So it's probably not some like one simple thing, but rather a combination of factors. I found a really great free text I will link to. It is live, which means that it's a living text, so they update it periodically. And it's got a lot of really great articles, and so I'm really enjoying reading it. And so hopefully you'll find it helpful too. And hopefully all of this was interesting to you.